Welcome. Today we're diving into hemoglobinopathies, the group of conditions that come from changes in the hemoglobin molecule. We'll walk through the hemoglobin molecule itself, practical diagnostic approaches, sickle cell disorders, thalassemia syndromes, hereditary persistence of fetal hemoglobin, other variant hemoglobins, and finish with some practice exercises. If you've ever wondered why a tiny change in one gene can alter how blood carries oxygen, stick around. By the end, you'll understand the tests doctors use and what they're looking for. Start with the genetics. The protein parts of hemoglobin, the globin chains, come from genes on two chromosomes. The alpha genes live on chromosome 16 as two genes called alpha 1 and alpha 2. The beta family, including the beta and delta genes, sits on chromosome 11. Knowing where these genes are helps explain why different mutations affect different chains and why some diseases run in families. Blood expresses different hemoglobins at different times. Embryonic forms include Gower1, zeta 2, epsilon 2, and Gower2, alpha 2, epsilon 2, and two Portland types, zeta 2, gamma 2, and beta 2. Fetal hemoglobin, written HbF, is alpha 2, gamma 2. Adult hemoglobin, HbA, is alpha 2, beta 2, and the minor adult form HbA2 is alpha 2, delta 2. The point is the chain combinations change during development, and that affects oxygen affinity and clinical presentation. Hemoglobin problems fall into a few main categories. Some are structural variants, a changed amino acid producing a variant hemoglobin. Others are problems of reduced or absent synthesis, which we call thalassemia syndromes. And then there are cases where the normal switch from fetal to adult hemoglobin doesn't finish. That's hereditary persistence of fetal hemoglobin, or HPFH. Each category causes different lab patterns and symptoms, so the diagnosis depends on matching the clinical picture to specific tests. When a clinician suspects a hemoglobinopathy, they start with history, exam, and ethnic background, because some conditions are more common in certain populations. The basic labs are a complete blood count and blood smear, which can show anemia, small red cells, or abnormal shapes. For specific identification, we use hemoglobin electrophoresis on cellulose acetate at alkaline pH, electrophoresis on citrate agar at acidic pH, and high-performance liquid chromatography, or HPLC. Each test gives different complementary information. I'll explain how in the next slides. Remember a key physical fact. Hemoglobin molecules carry electric charge, so they move in an electric field. On alkaline cellulose acetate electrophoresis, hemoglobins migrate toward the anode, and typical bands you might see include AFS, or DG Lipor, and C, or EO Arab H. Note that HbA2 often appears in the same region as HbC in some systems. In plain terms, band position helps us narrow which variant or combination we're dealing with, but other methods are usually needed to confirm. On cellulose acetate at alkaline pH, the hemoglobin binds to agarroprotein in a way that makes the complex move toward the positive electrode. Any hemoglobin not complexed can actually be carried the other way by endoosmotic flow, a subtle lab detail that causes some bands to appear differently. High-affinity hemoglobins and certain variants can give unusual migration, so technicians often run additional media like citrate agar or agarose gels when results are unclear. Running electrophoresis on citrate agar or agarose at acidic pH changes how charge groups interact, so variants that look similar on alkaline runs separate on acid runs. That's useful because acid gels let us quantify normal and variant hemoglobins more reliably. For example, we can accurately measure HbA2, which helps diagnose beta thalassemia trait. In short, if alkaline electrophoresis raises a question, the acidic medium usually helps resolve it. HPLC, high-performance liquid chromatography, separates hemoglobins into distinct peaks. Important checkpoints when reading HPLC are a flat baseline, correct peak shapes, and the expected order of peaks, typically F fetal, then P2 and P3, minor peaks, then the main adult peaks A0 and A2. Total peak area is often in the range of 1 to 3 million units on the instrument, and HbA2 and HbF responses should be within expected ranges. HbA2 retention time commonly centers around 3.65 minutes with a small tolerance. HPLC is powerful because it both separates and quantifies hemoglobins. A quick screen for sickle hemoglobin is the Sickle solubility test. Blood is deoxygenated, historically using 2% sodium metabosulfite, and the sample is sealed under a mountant. If HBS is present, it polymerizes and makes the solution turbid or cloudy. If no HBS is present, the solution stays clear. The test can be fast, but it's a screen. A negative result may need confirmation after 24 hours in some protocols, and it won't distinguish trait from disease reliably, so follow-up tests are needed. A few other simple lab tests still have diagnostic value. The Kleihauer-Betke stain highlights fetal red cells. Fetal cells stain deep pink while adult cells are paler. If the staining is pancellular, it suggests HPFH, while heterocellular staining points to a thalassemia pattern. For alpha chain deletion diagnosis, we use the HBH prep. Exposure to a mild oxidant like brilliant crescent blue causes characteristic inclusions. Before splenectomy, you might see a golf ball appearance, while after splenectomy, you can see numerous Heinz bodies. These are quick, visual tests that provide strong clues. When initial tests don't give a full answer, we move to advanced methods. DNA analysis for specific mutations, globin chain synthesis studies, globin chain electrophoresis, and immunoassays for variant hemoglobins. Other tools include heat stability testing, isoelectric focusing, microcolumn chromatography, and capillary electrophoresis. Put together, these methods let a laboratory identify the exact variant or thalassemia genotype behind a patient's abnormal blood results.